For this section of the immune system chapter, we're going to consider adaptive immunity and focus on the lymphocytes. In addition to innate immunity, there's also adaptive immunity. The two together, of course, make up the immune response in its entirety, but the adaptive component is how we respond to specific threats, how we adapt to the environment around us. There are two components to adaptive immunity. There's what's called cell-mediated immunity and antibody-mediated immunity. These depend on the two different types of lymphocytes that we're going to consider here. Cell-mediated immunity is all about T cells and how they interact with other cells, whether it's the cells that remove pathogens from our system, namely the phagocytes, or the cells that are infected by those pathogens. And then antibody-mediated immunity is based on B cells, which produce antibodies and secrete them into the plasma so that they're found in our various bodily fluids. Let me show you this picture to remind you how the different types of blood cells are made in the red bone marrow. The hemocytoblast, the multipotent stem cell, divides, giving rise to a cell that will either become a myeloid stem cell or a lymphoid stem cell. The myeloid stem cell gives rise to all of the other types of blood cells, platelets, red blood cells, and the white blood cells that are not lymphocytes. And the lymphoid stem cell gives rise to the lymphocyte. Now, there's different types of lymphocytes, and we're concentrating in this section just on a couple of them. And those are the T lymphocytes or the B lymphocytes, the T cells and B cells. To understand adaptive immunity, we're going to consider the lymphocytes and their life cycle. So we'll begin with how they mature, from when they're first made in the red bone marrow, as the previous image showed, up until they're ready to go and perform their duty as part of the adaptive immune system. This explanation is going to concentrate on the T cells, which can also be called thymocytes, and I'll explain how the B cells fit into it a little later on. So all cells, B cells, T cells, and other blood cells, are made in the red bone marrow. The T cells are called T cells because they migrate from the bone marrow to the thymus to go through this maturation process. And the first step of that is what's called positive selection. Now, the T cells at this point are not very differentiated, so they're designated here as CD4 minus, CD8 minus, or negative, meaning that they don't express these proteins, which will become more important later on. They're tested first in what's called positive selection to be sure that they can interact with other cells of the immune system. What's very important is that T cells interact with other cells that express a protein on their cell surface called MHC. And so positive selection is looking to see that T cells can, in fact, properly interact with MHC. There's actually sort of three levels of binding. This picture only shows two, which is they bind or they don't bind. But it's also possible that T cells will bind to MHC too strongly and not let go, which is just the same as not binding at all as far as this is concerned. So when the T cells properly interact with MHC, they survive this round of testing, so to speak. And if they don't bind or they bind too strongly, they're destroyed. After completing positive selection, the cells that survive then go on to start expressing these two proteins, CD4 or CD8, which is part of their next phase. The next phase is called negative selection, and this is where the cells are tested to be sure whether they recognize self-antigens or not. We don't want cells of our immune system to recognize self-antigens because then we'll react to things that are supposed to be in our body. So this is negative selection, so that the cells that do not recognize self-antigens survive, and the ones that do react to them are destroyed. Now, this is also happening to B cells in the bone marrow, but again, we're concentrating on T cells in the thymus right now. And the point of this is to make sure that we don't have autoimmune diseases. So at this point, the T cells have passed both positive selection and negative selection, so that they properly interact with MHC and they do not interact with self-antigens. They then differentiate into CD4 positive and CD8 positive cells. They move from the thymus out into secondary lymphoid structures. The CD4 positive cells are going to become helper T cells. The CD8 positive cells will become cytotoxic T cells. They just have to learn about the environment at this point. So they're labeled here as mature cells, which means that they've finished going through this maturation process. But they're actually still naive. It's like they've finished college and they've graduated, but they don't know really about the real world. So they have to start doing their real jobs. This summarizes the process of that maturation. All lymphocytes are born in the red bone marrow. The maturation takes place in the primary lymphatic tissue. That's the bone marrow for the B cells and the thymus for the T cells. T cells go through positive selection and negative selection. B cells only go through negative selection. Positive selection is ensuring that they can interact with MHC. If they do, they get a survival factor and they continue on. If they don't get that survival factor, then they are destroyed. And then cells that interact with self-antigens are induced to die through a process called apoptosis. Apoptosis is a process of natural cell death. It happens in a lot of different developmental processes. One good example of that is in the embryo as the hand develops. There's tissue between the fingers, webbing, which would lead to a flipper in certain organisms. But for land animals, the tissue between those digits usually dies away through this process. Now the word is pronounced apoptosis. Apo, the prefix meaning apart, tosis means to droop or to fall. So it basically means falling apart. What happens is the cell sort of breaks down into smaller and smaller pieces that can then be phagocytosed by white blood cells to clean up easily after the dying cell. One way to think about apoptosis is based on an incorrect pronunciation of the word, which is apoptosis. It sounds like in that word that there's no popping, apoptosis. And you can think of that as a false etymology in when cells don't lyse, when they don't burst open, then you can easily clean up after the cellular debris. And the other type of cell death that we usually associate with the immune system is lysis, such as with complement and other effects that white blood cells will have on diseased and damaged cells. An important type of cell that interacts with the T cells for adaptive immunity is called an antigen-presenting cell. Now, antigen-presenting cells can be a few different types of cells, and they generally fall into two different categories: those that present exogenous antigens and those that present endogenous antigens. Exogenous means from outside the body, and endogenous means from inside the body. Exogenous antigens are pathogens that have gotten into your system and will cause infection. Endogenous presentation is referring to antigens that are being made by body cells. Self-antigens fall into that category, but really because of the negative selection process described earlier, they aren't a big player. Endogenous presentation is usually showing us when cells are doing something incorrectly. Cancer cells will start making certain proteins that they're not supposed to, or when a cell's infected with a virus and it's making new virus particles, those new virus particles are considered endogenous antigens also. Remember this picture that showed the process of phagocytosis. At the end of this, the pathogen has been broken down into debris, some of which has been released from the cell and some of it is retained by the cell. What's retained in the cell is then going to be presented on the surface of the cell to indicate to T cells presence of those particular pathogens. This figure from the book sort of completes that story. We actually see both endogenous and exogenous presentation here. So at the bottom, next to the section that says number 1 here, a pathogen's being ingested into this cell to be broken down. And what's left over of that is going to be paired with type 2 MHC and presented on the surface of the cell. That's exogenous presentation. The pathogen's been digested by the cell and presented on its surface. Endogenous presentation is represented a little bit oddly here because we're looking at a dendritic cell. But what happens in endogenous presentation is antigens from cells in the body are presented on the surface of the cell using type 1 MHC protein, and those are going to interact with the cells. Now, because the book is using dendritic cells as an example of phagocytic cells, I want to use a different picture to illustrate these two processes a little bit better, because dendritic cells' involvement in an endogenous presentation is a little unusual. It's more about the cells themselves presenting things on their surface. So let me use a different picture to show that. This image is from a biotechnology firm that's demonstrating the different types of antigen presentation. And on the left side of the image, the exogenous presentation seems to be a little bit more involved than what the book has shown. They're just highlighting another protein that's part of the process for the sake of what that company uses. But the idea is still the same, that something from outside the body has gotten in, and a phagocyte brings that into the cell, digests it down in the phagolysosome, and then parts of that are presented on the surface of the cell with MHC2. On the other side, the endogenous presentation is a little bit better represented for what happens in your average body cell. There's a structure called the proteasome, which breaks down proteins in the cell that have lived their usefulness in the cell or that might have problems with them, or those sorts of things. Those proteins will then be attached to MHC type 1 to be presented on the surface of the cell. The final step in the lymphocyte life cycle is to be activated. These are the naive cells. They've moved out into the secondary lymphatic tissue, and they're going to be interacting with the antigens that they're meant to deal with. First, we want to see what happens when the CD4 positive cell interacts with an antigen presenting cell. Now, this is going to be a phagocyte presenting a processed antigen, an exogenous antigen on its surface with MHC2. CD4 interacts with MHC2. CD8 will not. CD4 does not interact with MHC1. So that has to happen, and then the T cell has a receptor that recognizes the specific antigen. And when that happens, the two together causes activation of the helper T cell. There's a signal that's released from the antigen-presenting cell that actually causes the activation of the helper T cell here. And when the helper T cell is activated, it's then going to start releasing cytokines to help activate other cells, which is where the name helper T cell comes from. CD8 positive cells, which are going to become cytotoxic T cells, interact with antigen-presenting cells that present with MHC type 1, and they have their T cell receptor that recognizes the antigen. Now again, CD8 recognizes only MHC class 1, and then the T cell receptor recognizes the antigen. Those two things have to happen for activation to take place. Now in the pictures from the book, using dendritic cells as an example, the dendritic cell would release a signal that would help to activate the cytotoxic T cell. In situations where the antigen-presenting cell can't do that, if it's a cancerous cell or a disease cell or something like that, it might not be able to release the co-stimulation factor. And so helper T cells releasing cytokines are there to help to activate the cytotoxic T cell. When cytotoxic T cells become active, they proliferate and differentiate. They become multiple copies of the same cell, which are all directed at the same antigen. And they differentiate into the active cytotoxic T cells and memory T cells, which will persist to help fight off future infections of the same thing. Now, helper T cells actually do the same thing. The previous picture didn't indicate it, but there are helper T cells that are actively releasing cytokines and memory helper T cells for future infections. Though we've been concentrating on the T cells, I want to include B cells in the activation process. They're a little bit more simple because they don't have antigen-presenting cells interacting with them. Instead, a B cell receptor, which is a special type of antibody, on the surface of the naive B cell interacts with the antigen, and then it gets activated. Now in this picture, they jump from antigen binding to the B cell receptor to the helper T cell helping to activate the B cell. What's happening in between, where that arrow is the antigen gets internalized and then presented on the surface of the B cell through MHC2, so that CD4 positive helper T cells will recognize that, and the proper helper T cell will recognize that antigen and help the B cell become activated. Now, we're going to look at what happens with antibodies in a little while, but before we go into that, I want to finish this section by looking at a very nice video showing how cytotoxic T cells perform their function and destroy cells. It's going to concentrate on cancerous cells, but the same thing could be said of diseased or other damaged cells in the body.
Inside all of us lurks a serial killer. A killer whose primary function is to kill and then kill again. These are cytotoxic T cells, a specialized member of our white blood cells. They patrol our bodies, identifying and destroying virally infected and cancer cells. And they do so with remarkable precision and efficiency. There are about 5 million T cells in a teaspoon of our blood, engaged in the ferocious and unrelenting battle to keep us healthy. These amorphous blobs move around quite rapidly, pushing out their leading edge and probing their environment as they go. When a cytotoxic T cell finds a cancer cell, membrane protrusions rapidly explore the surface of the cell, checking for the telltale signs of cancer. They kill their targets using poisonous proteins visible here in red. These cytotoxic granules move down special pathways in the cell called microtubules to the interface between the T cell and the cancer cell. The T cell punctures the surface of the cancer cell and delivers its deadly cytotoxins. This is very important in our bodies, where cells are packed together, as it focuses the lethal hit on the target and minimizes collateral damage to the neighboring healthy cells. The fate of the cancer cell is sealed. The T cell then moves on, hungry to find another victim.
Consider this question. When you feel that you've reached an answer, hit the next button to go into the next slide, which will reveal the correct answer. This question might have been a bit intimidating, trying to figure out all these things together, but it's actually fairly simple. Just remember that CD4 cells, which are helper T cells, interact with MHC2. CD8, which are cytotoxic T cells, interact with MHC1. And the only place where we see that connection is in the first option. CD8 can't be helper T cells, CD4 can't be cytotoxic T cells, and CD8 can interact with MHC2. So the only thing that actually puts all the things together correctly is the first choice. And in fact, phagocytes will present exogenous antigens to CD4 positive helper T cells using MHC2.